Welcome. We're going to begin talking about the endocrine system today. So this will take a few times to get through all of the slides for our endocrine. So the endocrine system is one of our main ways that our body has to communicate. And so way back in A&P1, we talked about kind of the systems in general, and the other way that we communicated was through the nervous system. And some of the differences between the two, the endocrine system is relatively slow for the most part to get its message to where it needs to go versus the nervous system is very quick. The nervous system tends to work in kind of the momentary versus the endocrine system working in kind of more the long-term effects of the body. The nervous system is there for the moment-to-moment changes. The endocrine system working along the lines of making sure that things are good for the long haul, so to speak. Some of the other differences come in the way that the systems are set up. With the endocrine system, there's no kind of centralized area. So we have endocrine glands that are found throughout the body itself. Different organs, different tissues are going to be able to produce uh, hormones as well. Some in larger components, some in lesser components that are there as well. We have others that have kind of dual function. So we have things like the pancreas, where we have both an exocrine and an endocrine function. And then we have other things like the gonads, which are there for both reproduction and the production of hormones as well. And that's there. When it comes down to the endocrine system, though, the hypothalamus is kind of our master control. This is the place that's going to kind of dictate over everything and be the beginning point for the most part for all of our structural components as far as beginning the hormone cascades that we have. And as I said, other tissues do have the potential to produce hormones. A lot of these hormones are going to be local hormones working within the organs themselves, things like that. We do have some that are going to be a little bit further in terms of their kind of uh, production in a sense as far as where they're going to go. Things like the kidneys, the heart have kind of far-reaching effects because they're dealing with more kind of global issues as well. So here we can see the distribution of many of those endocrine glands. They are spread throughout the body in many different places. So they're not in a centralized area, which means that the other side of the equation, so to speak, for the endocrine system is that it doesn't have a direct pathway to get its hormone to the individual organs that it targets. And so it has to utilize the cardiovascular system in order to send those substances along. That being said, the hormones themselves are similar to that of the nervous system's use of neurotransmitters. So we're going to have some sort of chemical that's going to go out and it's going to find its target organ that is there, and then it's going to bind to a receptor once it gets there. In that process, the cells themselves distribute their hormones out into the extracellular fluid. So they don't have a duct by definition, the endocrine glands. They're simply secreting into that extracellular fluid. And then from there, they're going to get picked up by the cardiovascular system. So they're going to end up into the veins and into the system there, which leads to us having quite a bit of a lag time at times. And certainly this can be just simply a few seconds, but this could be even hours for things to start to get to have any kind of an effect that is there. Once they do start, we tend to have a prolonged effect. It's going to take a while for those hormones to kind of kick in, so to speak. And then once they're there, we're going to make it work for minutes, hours, days, weeks at a time. And so depending upon what that particular type of a hormone is designed to do. Most of them are relatively long range hormones. They're going to go from one of those distant organs all the way out to its target organ somewhere else in the body. Others are kind of more locally acting. So we have things like eicosanoids in terms of hormones that are going to act typically right in the tissue that they are found. When it comes to hormones, just like just about everything else in the body, we kind of divide things up in the body itself into classifications. In this case, we did it with the neurotransmitters as far as the type of neurotransmitter that we had. And now we're going to do the same thing here for hormones, and that we're going to divide them up based upon kind of what makes up the hormones themselves. And so we have lipid-soluble kinds, Um, these tend to be steroid-based types of hormones. They're going to have things like our thyroid hormone that is there. And then we can even have some other novel types of hormones, things like nitric oxide that can work as well. Lipid-soluble hormones, obviously they're lipid-soluble, so they're going to be able to go through plasma membranes directly, versus water-soluble hormones, they're going to have to stay on the outside of the cells, bind to the receptors there, or bind to a second messenger system, something like that. And so we have things like our amines, we have peptides. These are protein-based substances or amino acid type of hormones that are there. And then we also have our eicosanoids. And so those eicosanoids are the more locally acting types of hormones. And when they are locally acting, they tend to be either what we call autocrines or paracrines. And so autocrines basically stimulate themselves. And so they're going to release a hormone that's going to have an action on its own self. And so that hormone feeds in onto itself, versus a paracrine is going to affect other cells. And so here we have an autocrine down here at the bottom. It releases its hormone and it binds to receptor on its own cell. Versus a paracrine, it releases it and it goes out to another cell, but that target cell is close by. It's not very far away from that. And so these eicosanoids, these locally acting hormones, are typically in the tissues they are. This may be in something like the digestive tract, where we're just going to kind of go a relatively short distance. Others kind of are more typical hormones, are more of our circulating types of hormones. They're going to go out into the extracellular fluid and then make their way into the blood vessel and then travel to long distances over to our target organ, someplace else in the body. Once hormones get to their target, they're going to need to bind to some sort of receptor. And so this can happen in one of two ways. They can either be direct acting types of hormones. And so we have direct activation. This is going to directly turn something on, turn something off in the cells. Think of like kind of our acetylcholine and skeletal muscle. As soon as that acetylcholine bound to the outside of the cell, we had a direct effect on the cell. In this case, generally it's going to have some sort of gene activation rather than kind of just necessarily opening up an ion channel that is there. And then we can also have those second messengers that are there. And so second messengers I mentioned under the water-soluble types of hormones, and that's because they're going to bind to the outside of the cell, and they've got to make some sort of change inside the cell, but those hormones can't get in. Versus our direct acting, those are lipid-soluble ones. Those can slip through the plasma membrane, make their way to the inside of the cell, and they're going to directly bind to a receptor, and that's going to cause some sort of change inside the cell in terms of a gene activation that's there. Second messenger proteins need to have that G protein linked type of receptor. So they're going to bind to a G protein linked receptor, and then we're going to have that cascade of chemical reactions that leads us to our second messenger that is there. Depending upon the hormone, a single hormone in general may go out and have more than one type of receptor. And so when it goes out there and starts to bind to those different things, depending upon the type of receptor that we have, in this case I just did A, B, and C, that particular type of receptor is going to lead to a particular type of response. And so that could be to, in a sense, turn something up. It could be to turn something down. It may be, in a sense, to cause a reproductive process. So kind of an arrow with a circle there. We're going to reproduce something inside the cells, turn something on, turn something off. But it all depends upon the particular protein, the particular receptor that is on the target cell itself. If the target cell has one particular type of a receptor, we get one response. If it has a different type of receptor, we get a different response. And so it's very target specific, and it's very receptor specific as to what kind of response we're going to get in those particular cells themselves. So once we get there, And we saw this before, again, with things like muscle. In muscle, we had acetylcholine, and when acetylcholine bound to skeletal muscle, it made it active. It caused it to contract. But when acetylcholine bound to cardiac muscle, it caused it to become inhibited. And so it caused it to have more relaxation. So same chemical substance, different receptors, different effect. That's there. And so they can have all kinds of effects on the cells themselves. We're familiar in the past with things like our plasma membrane permeability changing. Think of skeletal muscle and or muscle in general and nervous system. We can turn things on in terms of protein synthesis. We can activate enzymes or deactivate those enzymes in a particular system. We can cause something else to become secreted. And so we can cause secretory activity. We may go from, say, one hormone down to another gland, and then that other gland gives us another hormone. And then that finally gets us to our true target in the system. And so that target then is the final product, so to speak, that's there. It can turn on cellular reproduction. All kinds of activities can happen with those hormones. Again, all dependent upon what the receptor is and how that hormone is going to act on that particular receptor. We'll take a look at the individual hormones themselves as far as the general classifications, and then we'll start to take a look at the specific hormones that we have in the body. So lipid-soluble types of hormones are those that are either steroid hormones, thyroid hormone. These hormones are going to get through the plasma membrane. They're going to go into the inside of the cell, and so once they're in the intracellular space, they're going to bind to a receptor that is inside that intracellular space. So it's inside the cell at this point, and the hormone and the receptor now form a complex. This complex is going to go from where we were now just inside the cell, and we have our hormone, and we have our receptor. This is now going to travel into the inside of the nucleus, and inside that nucleus, we're going to then cause some sort of change. And so we're either going to have some sort of interaction that is going to potentially cause something like the transcription of DNA. So we're going to produce mRNA. Now from that mRNA travels back out into the intracellular space that's there, and we then start to produce proteins of some sort. In this particular case, that's there. And so we're manufacturing some sort of substance. That substance may even be another hormone. And so that's going to move then to the ribosomes. Ribosomes are going to make our proteins. Proteins then get sent to wherever they need to go from that point, whether it's over to the Golgi apparatus to get packaged up, whatever it happens to be for that particular cell. So comes into the hormone itself, comes in to the cell, makes its way into the intracellular space that's there. It's going to bind to a protein. In this particular case, that protein, that receptor itself actually also happens to be inside the nucleus. Once it's there, it can then start to form a change on the DNA, and we start to then produce our mRNA. mRNA goes over to a ribosome, makes our new protein, and whatever that new protein is, it then does its function in the cell. That's there. And inside that cell, then we may either make new proteins, package it up inside the cell, send it out wherever it happens to be. Different picture, same concept here. So we have our protein receptor complex inside the cell itself. That receptor protein complex, once they bind together, we now have the ability to come into the DNA, bind to the receptor on the DNA itself, and then once again manufacture our new protein. Now we have our new protein, package that up, send that out, or utilize it inside the cell for whatever it is that we made it for in the first place. For our water-soluble types of hormones, those water-soluble ones are going to bind to the outside of the cell. So this is going to utilize a second messenger system. And so the first messenger is the actual hormone itself that came along. It binds to a receptor on the outside of the cell. But inside the cell, so we have the plasma membrane here, we have, say, our receptor of sorts, and then we have our hormone that's going to come along and bind into that receptor. On the inside of the cell, we've got another mechanism. That other mechanism is our G protein that is on the inside itself. And so we have a G protein linked receptor. That activates our G protein on the inside of the cell, and this is then going to start a cascade of chemical processes inside the cell itself. And so we now have our activated G protein. This is going to activate the effector enzyme adenylate cyclase. So we have our adenylate cyclase now is turned on. We have our next enzyme in the cascade, so to speak, on there. And this is going to eventually lead to the conversion of ATP into cyclic AMP or CAMP. And so we have, cyclic AMP in there. And so our cyclic AMP now becomes our second messenger. This could also be the activation of calcium inside the cell. Other things, we have other types of second messengers as well, but cyclic AMP is a biggie. We could also have cyclic GMP as a particular second messenger as well inside the cell. Either way, we've activated these second messengers. And the second messenger now is going to travel inside the cell, and it's going to activate or inactivate cellular function. It's going to turn things on, turn things off, whatever it happens to be inside that cell itself. Once we've activated that cyclic AMP, we don't want to have, in a sense, turned on all the time. So it's going to become inactivated by phosphodiesterase. And so we have another enzyme that's going to cause that cyclic AMP to become inactivated, and we can move on, in a sense, to the next time period. We may be looking for more hormone to come along to tell us that something is going to come along the way at that point. So our little hormone here comes along. So we've got our water-soluble hormone. It's going to bind to the receptor. That receptor itself has the G protein there. And when that receptor binds to the protein, it's going to activate our G protein. G protein then, with its activation, leads to the conversion of ATP into cyclic AMP. Cyclic AMP is now our activated second messenger. Our second messenger is then going to activate protein kinases. And so those protein kinases are then going to go out and lead to other processes inside the cell itself, and so activate those other processes. In order to turn off this cyclic AMP, we again have our phosphodiesterase, and that's going to lead to the breakdown of our cyclic AMP as well. The fact that we activate protein kinases inside the cell means that we could end up having a huge response in a sense. And so we can have from essentially this one little hormone, we could have potentially millions of activated enzyme. And so these reactions now are leading to the potential long-term activation inside the cell of whatever particular responses that had inside the cell itself. To a certain degree, kind of like with the nervous system, we have specific tissues that we have as a target. And so the target tissues themselves are going to only be responsive to that particular hormone itself. So target cell is what we're looking for. That target cell is very specific in terms of that we're looking for a particular receptor. And so we have a receptor on the outside of the cell. Here we have our cell. Here we have our receptor. And here we have our hormone. If that hormone comes along, it can go in and bind to that receptor because it fits. But if that particular cell that we have has a different kind of receptor, and now our same hormone comes along, that hormone can't fit into that receptor. It doesn't fit there. So it's going to then have the components that are there for the ability to receive that or not. In addition, any given cell, our target cell, is going to have a number of receptors on the target itself. The more receptors it has, the more, in a sense, kind of susceptible, so to speak, or the more sensitive it is to that particular hormone. So if we increase the number of receptors, we have an increase in sensitivity. Just erase those there. So our sensitivity goes up. If we have a decrease in receptors, this is going to lead to a decrease in sensitivity. Meaning that the cell is not going to be as receptive to that particular hormone. So that is what we're going to have there. We can also have a change in the affinity of those receptors. So they may want to bind that hormone. They may not want to bind that hormone. They may have a greater or lesser affinity for them. So that kind of increase or decrease in receptors is what we get when we have downregulation and upregulation. And so if a particular target cell has more receptors, that's going to lead to an increase in sensitivity to it. And so this may be a situation where the particular cell hasn't really been stimulated by that hormone very much. So it goes along and it's kind of, in a sense, lacking stimulation from that receptor. So it goes and it adds a whole bunch more receptors. In downregulation, we take from, in a sense, all of those receptors, and now we're going to make it so that we have many less. This is kind of the concept behind what happens when we have something like type 2 diabetes. And so the idea behind there is that we have insulin that is being constantly secreted into the bloodstream, and it's going out and telling the cells, hey, take up this glucose, take up this glucose. And the cells kind of get tired of it, so to speak. And so they downregulate the number of receptors that are there. And so they have insulin resistance. They're no longer responding to that insulin that's there. They have normal, if not increased amounts of insulin production, but they don't have the ability to bind them there anymore. And so these things kind of change at different times within there. Hormone concentrations in the blood also leads to the potential for us to have a response or no response to the particular hormone itself. And the amount of concentration that we have in there is going to be related to how much are we releasing, so what's the production of that endocrine organ, and how quickly are they becoming inactivated in the body. So in terms of our breakdown in the body, this can be happening because we have enzymes that are floating around in the blood. So they may be getting broken down in the blood itself. They may be lost as urine, or they could be being broken down by liver enzymes themselves. And so this is going to cause the breakdown of our hormone. And that's going to dictate how much of that hormone we actually have in the bloodstream itself. If things are getting lost in the urine or they're getting broken down by the liver too much, our hormone levels may be declining. And on the converse, if they're not getting broken down, if they're not getting lost, we may have increased levels of those hormones going up as well. So it's this balance within the body of kind of regulating our hormone levels around kind of a mean that we want to have. And so we have that level that we want to control them there. And this leads us to our negative feedback system. And so things are going to allow us to increase the levels, allow us to decrease the levels, and they're going to be there in just a kind of a narrow range. And so we're not going to vary all that much as far as those total levels go. Things go up, we want to bring them back down. They come back down, we bring them back up. And so they're constantly going through this process. And in order to kind of determine where or how much or when we need to turn things on or off, we have three different types of control. We can use chemical control, we can have neural control, and we can have hormonal control. And so three different processes there that can allow us to look at the response of that endocrine organ based upon one of those three components. And so we have humoral control. This is going to look at the actual secretion of the substances that are there based upon things that are in there. So this is our type of control based upon what's inside the blood itself. And so calcium ions we looked at back in bone, are being dictated in their control based upon both parathyroid hormone as well as calcitonin. And so we have two hormones to kind of control things going up or down in terms of calcium in the blood itself. So if we have a loss of blood calcium, this is going to stimulate more parathyroid going out. More parathyroid hormone going out is going to cause those calcium levels to start to rise. And so we're going to have osteoclastic activity increasing. We're going to absorb more from the gut. We're going to reabsorb as much as we can from the kidneys and so on. And we're going to bring all of those substances back into the body. But once those levels go up too much or get risen back up into the normal, then calcitonin levels are going to go up and parathyroid levels are going to go down in that particular case. And so we're having them go up and down based upon the rise and fall of ion levels or nutrients in the blood itself. Same thing would go for, say, glucose levels. And so glucose levels, as they rise, are going to lead to more insulin being released. As they fall, insulin release goes down. And at the same time, glucagon levels are going to start to rise. And then as the blood glucose levels rise, blood glucagon levels are going to fall as well. That's there. We can also have direct neural control, and so the nervous system itself can be the control for our system. And so the nervous system can stimulate our endocrine organs themselves. We'll see this when we get into the posterior pituitary gland and the hypothalamus. We'll see when we get into the adrenal glands that we have this concurrent nervous control that helps with the release of the hormones themselves. And then lastly, we have hormonal control. Hormonal control is going to work on the level of the individual hormones themselves being risen or lowered to dictate whether we get more or less of that particular hormone. And so these are things like thyroid hormone, and so which is going to be involved in a whole cascade of our thyrotropin releasing hormone, our TSH, and then our thyroid hormone kind of controlling there as we raise or lower those levels within the blood itself. So three different ways of control. And when we start up next time, we'll talk about the individual hormones themselves.